All right. All right, well, I'll go ahead and start with the introductions. Welcome to this inaugural PMedic seminar. This is the first installment of our new quarterly research seminar series. PMedic is a joint institute of Oregon Health and Science University and the Pacific Northwest National Laboratory. I'm Beth Habecker, the OHSU co-director of PMedic, and it's my pleasure to welcome you this afternoon. The goal of PMedic is to leverage the strengths of both institutions through collaboration, integrating discovery science and research with clinical practice in order to improve human health and disease treatment. We'll be hearing today about one of those collaborations. After introducing the OHSU speaker, I'll hand things off to my counterpart, Bobby Jo Webb-Robertson, who will introduce the PNNL half of this collaboration. Dr. David Kohler is a professor of molecular and medical genetics at the OHSU School of Medicine. He received his medical degree from the University of Illinois, did a residency at the University of Minnesota, and then did a research fellowship at the National Institute for Child Health and Development in Bethesda, Maryland. He has a research background ranging from very basic studies to human epidemiology. Most important for our purposes today, he is the OHSU co-PI of the Metabolomics Core for the NIH Undiagnosed Diseases Network. That is the work he will be talking about today. Dr. Webb-Robertson will now introduce us to the other co-PI.
Thank you, Beth. As Beth mentioned, I'm the P&L co-director of our PMedic Collaborative Institute between the two universities, or the two institutes. I'd like to introduce Dr. Thomas Metz. He is the Metabolomics team lead in the Biological Science Division at PNNL. He did his PhD at the University of South Carolina in chemistry and a postdoctoral fellowship at PNNL in mass spectrometry. And he stayed on here after that postdoc. And since that time, he has spent his career studying the role that molecules play in promoting and resolving disease, mostly through advanced mass spec-based metabolomics, lipidomics, and proteomics. Again, for our purposes today, he's the P&L co-PI of the Metabolomics Core for the NIH Undiagnosed Disease Network. So the last thing I want to say before I hand it off to Dave and Tom is a reminder for those who joined a little bit later, the meeting is being recorded. And as such, we would appreciate if you put your questions in the chat, and then we can either call on you or read them at the end of the presentation today, rather than interrupting Dave and Tom during the presentation. Thank you, Dave.
Okay, thanks to both of you, and welcome everyone to this inaugural discussion of collaborations with PNNL. I'm going to go ahead and share my screen here. Hopefully, there we go. And just to confirm that's working, people are seeing a slide there.
Yes,
yep.
Looks good.
All right, okay, great. So as you know from the flyer, what we're going to talk about is the metabolomics in terms of the role in characterization of rare diseases. And the way we're going to do this today is to really kind of talk about what is the Undiagnosed Disease Network in terms of what its history is and what the goals of that program are, and then and I'm going to cover this part, and then Tom's going to talk a lot about the core in terms of what was our approach and how did we set about to use metabolomics as a diagnostic tool. And then I'll come back again and kind of go through some of the lessons learned, and then we'll leave plenty of time, hopefully, for some questions. What I'd like to do is just kind of an introduction to the Undiagnosed Disease Network is to actually begin with a clinical case. This is actually a story of a family that I was involved with about 10 years ago, but I think this helps to illustrate the the need and the role of something like an undiagnosed disease network. So this was a 2-year-old girl who presented with a recent onset of an unstable gait and some tremor. And she was initially seen at OHSU in the pediatric neurology clinic at about 2 years of age for this gait instability that had just begun several weeks earlier. Parents had also noted there was a recent onset of a tremor when this little girl would be reaching for objects. The past history was pretty unremarkable, normal pregnancy, delivery, had some issues with recurrent ear infections, but that was about it. There had been a recent viral illness associated with some fever and some vomiting about 6 weeks prior to the onset of these symptoms. The family history was pretty unremarkable as well in terms of no history of neurologic disease or ataxia, tremors, any other related kinds of conditions. And developmentally, this child seemed to be doing okay, initially sitting up at 8 months, crawling at 10 months, and starting to walk at about 14 months, and was walking age appropriate up to the time of the onset of these symptoms. The language development was somewhat delayed at 2 years of age with only 20 words and not using 2-word phrases, but could both point to some body parts. The exam was remarkable for a couple of things. First off was the growth was not very good in terms of the height being less than the 3rd percentile and the weight being only about the 5th percentile for age, whereas the head circumference was at the 50th percentile. And the neurologic exam was remarkable for this wide-based gait and truncal ataxia, and also the tremulousness noted by the parents. But otherwise, the muscle tone and the reflexes were okay, and the sensation seemed to be intact. A brain MRI study was unremarkable at this point in time. So the thoughts of the neurology team at that point was, well, it could have been something related to this viral illness, just a post-infectious ataxia, or the other big concern was a neuroblastoma potentially presenting this way. And so they brought this young girl back to the hospital for 24-hour urine collection with collection of catecholamine metabolites, CT analysis of the chest and abdomen, pelvis, looking for this potential neuroblastoma, and some routine lab screening tests. And all of these things were normal. So a month or so later, she was first seen by one of my colleagues, Dr. Harding, in the OHSU Metabolic Clinic, and the story was similar. The ataxia had continued, was falling frequently, and interestingly, they noted that this seemed to be a fair amount of fluctuation in the symptoms. So at the end of the day, when she would be more tired, the symptoms would get worse. Other than that, there had been no hospitalizations, no episodes of what we might call metabolic decompensation with acidosis, vomiting, dehydration. And then the other thing was there was no apparent decrease or regression with her cognitive development. It was just this motor dysfunction. So a variety of screening tests were done based on this evaluation. And once again, all these studies came back as normal. Some additional things at about 2.5, 24-hour EEG was done, which showed she was pretty much having continuous seizure activity, although these were not associated with clinical signs of seizures. Because of the concern about a mitochondrial disorder, a muscle biopsy had been done, and that was also unremarkable. Again, follow-up in neurology. There's been progression, so her gait has gotten even more unsteady. And at this point in time, she really couldn't walk without assistance and was using a wheelchair in order to enable her to get around on her own. One of the concerns because of the symptoms was that she had a form of a progressive myoclonic epilepsy. And so at this point in time, this was like 2010, there was a fair number of these genes known for this, and so a panel to test for these was sent, and once again, this testing came back as negative. The family subsequently had a variety of evaluations at Seattle Children's. They went to the Cleveland Clinic, and they went down to Texas Children's at Baylor. And all these studies and evaluations, people had various ideas and did various labs, but nothing came up. No diagnosis was ever reached. I sent some brain MRIs to a colleague in Amsterdam, and again, came back just generalized neuronal degeneration, nothing real specific. There was concern for a possible storage condition, and so we had recommended that she have a rectal biopsy, which was done, and by light and electron microscopy, there was no evidence for any sort of inclusions that we might expect in the types of storage disorders that Dr. Vandernap had suggested. So once again, we were clueless in essence. By the time she was about 8.5 years, her symptoms had continued to progress, and at this point, she had a younger brother, who I saw at about 2 years of age, because he was now showing evidence of global developmental problems. And when I saw him, his exams showed decreased muscle tone and decreased reflexes, and he also had some dystonic movements of his left hand and arm when he was reaching for objects. He was able to sit, but he couldn't walk, which is a significant impairment at 2 years of age. So at this point in time, I made a referral of this family to what was then known as the Undiagnosed Disease Program at the NIH. We sent the family off, and shortly thereafter, they did whole exome sequencing and identified homozygous mutations in both of these kids in the KCTD7 gene, which encodes a protein that functions as a functional role as a potassium channel. Now, this was a time when whole exome sequencing was not something available clinically. So this was a big deal. This was really a good result for us. And as it turns out, this family, this gene had been previously identified, and there had been one report of this from a large pedigree of a consanguineous family from Morocco that had a progressive form of myoclonic epilepsy, similar to what our patients had shown. And it was shown to be an autosomal recessive due to this KCTD7 gene. And turns out our patients were the first two that were not in that original family that were identified with this condition. So what is the NIH Undiagnosed Disease Program? Well, it was begun in May of 2008, and it was operated out of the clinical center at the NIH. This was really spearheaded by Bill Gall and Cynthia Tift, who were in the Genome Institute. And so patients were brought in, such as our family. They would get these thorough evaluations done at the clinical center. Of course, there was no cost and no need for insurance for these patients. And again, one of the key things at that point was that the NIH had technologies that we didn't have access to clinically, such as whole exome sequencing. So the Undiagnosed Disease Network, or the UDN, is really an expansion, if you will, of that undiagnosed disease program. And it started off in 2013, and the support came from what's known as the NIH Common Fund. And the goals of the program were similar to those of the Undiagnosed Disease Program, and essentially was to improve the level of diagnosis and care for patients with rare and undiagnosed diseases, facilitate research into the etiology of these conditions, and to try to develop an integrated and collaborative research community to identify improved options for identification and management of such patients. So this is a slide that just shows what the ultimate structure of the network was. Kind of go through this based on what you can see here from this key. So there were 6 clinical sites initially in the program. That's subsequently expanded. And these included 3 of the Harvard hospitals. There was a program at Duke, and then in the Midwest, there was Vanderbilt, and then at Baylor, and then out here in the West Coast, there was a site at Stanford and at UCLA. And all this was being coordinated by a coordinating center, which was also located at Harvard. They funded 2 sequencing cores, one at Hudson Alpha in Huntsville, Alabama, and then also in the genetics department at Baylor in Houston was a sequencing core. It included a central repository, which was being run by Vanderbilt, where samples from patients were being saved and stored for potential future research. And then they also developed a model organism screening core. And the primary site for this was once again in the genetics department at Baylor, but there was also a site at the University of Oregon where they were utilizing the zebrafish model. And then the other thing that was included was a metabolomics core. And this was based at PNNL and then of course at OHSU as well. And there was a couple of unique things in terms of the approaches to diagnosis. And one of those was the metabolomics core, because at that point, and really at this point in time, metabolomics wasn't really something used as a diagnostic tool. So it was really considered sort of a research aspect of the UDN to see what the potential role of metabolomics might be. And likewise, the Model Organism Screening Center, which again was one site at Baylor, where Hugo Bellin is a PI, and they developed primary Drosophila and mouse models, and at the U of O, where Monty Westerfield is the PI, and they developed zebrafish models. And the idea there was when novel genes are identified, and particularly genes that don't have a really known function, that they could use the core to generate animal models and try to better characterize what these conditions were all about. And then the other things that were unique was that they were using sequencing cores, and then some of the individual clinical sites also had technologies that, again, weren't typically clinically available, at least at the outset. And so that things like whole exome and whole genome sequencing, which of course are now readily available, but genome-wide methylation studies, transcriptome or RNA-seq analysis, and then more recently, what's referred to as optical genome mapping has been incorporated into some of the evaluations. And I think in particular, with regards to the DNA analysis, those of us who order a lot of whole exome sequencing know that there's an awful lot of things we can't pick up merely by that. And so things such as, in particular, the RNA-seq or other approaches to try to get at this issue of mosaicism have really turned out to be important. And I think particularly the RNA-seq is liable to become more commonly used, but difficult to incorporate into most clinical evaluations. Now, overall, the network included 18 institutions, which is kind of a beast to work with, but credit be given that we all worked together and there was network-wide protocols in terms of sample collection, processing, and other things. And a critical factor was there was a single central IRB, which was based at the NIH. And there's, of course, data sharing and use agreements that we were all part of so that all these this information could be shared. And the other thing that was a big part of the UDN, it remains an important part, is the idea of sharing the information that comes out of all these studies. So you've got diagnostic information such as DNA and other sorts of results that are put into databases. And then also clinical phenotypes were documented using the HPO or human phenotype ontology terms. And those were also, as appropriate, shared with various online databases. And I would just highlight one of those databases. It's called the Monarch Initiative. And we were very fortunate in that one of our partners in this, another OHSU faculty, was Melissa Hendel, who was one of the people who was instrumental in developing the Monarch Initiative. And so Melissa had actually already been playing a role with the Undiagnosed Disease Program on the bioinformatics side. And so she was a critical component of our proposal and of our team from the Metabolomics Core. And so now I'm going to hand this over to Tom to talk about the Metabolomics Core. And as I told Tom before, you really have to be careful about sending pictures to people, because here's the nice picture from Tom in the pre-COVID era that was on the flyer that everyone got for the seminar. And then he happened to share this with me, his COVID version. So with that, I will end my sharing and let Tom take over.
Hey, thanks, Dave, for an introduction that's really hard to follow. I can't follow that act. Let me get my slides going here. Can you see these?
Yep.
All right. So for those of you that may have attended previous PMedic seminars or are familiar with PMedic, the next couple slides may be already information that you've had presented to you, but I always like to start out with a little bit of at least geographical overview of where we are at Pacific Northwest Laboratory. But for those of you in the Portland area, we are about 3.5 hours driving time to the northeast, right about at the confluence of the Snake, Columbia, and Yakima rivers here in Tri-Cities, Washington, also known as Richland, Kennewick, and Pasco. And as a U.S. Department of Energy national laboratory, we have certain missions that we tend to focus on. Other laboratories in the national laboratory system tend to have different, sometimes complementary research foci, and you can see those listed in the upper right of the slide there. P&L, I had to update this slide just recently because I looked to see how many staff we now have, and we have 4,997 staff as of the last count, and so I put approximately 5,000 there, and business volume of about 1.1 billion, and that is spread over a number of different funding agencies, NIH, Department of Energy, Homeland Security, and other sponsors. Importantly, also for this presentation, we have a number of scientific facilities here, and that includes the EMSL, the Environmental Molecular Sciences Laboratory. You can see the photo here, and that is a U.S. Department of Energy National Scientific User Facility. And everything I'm going to talk about going forward in terms of the analytics that were employed for the UDN Metabolomics Core are present in this user facility and occurred in this building during the course of our studies of these individuals with rare disease. Also, I'd like to give an overview of the group that we're in here at P&L, which is the Integrative Omics group. And this is a group that has been around in one form or another for probably 30 years. And in our current form, we've been going for a little over 20 years. And that current form happens to coincide with when the EMSL was established as a building, as well as when the user program was established by the Department of Energy. And the Integrative Omics group has focused through the years on the development of instrumentation, software methods, and then application of those in the study of biomolecules, usually proteins, metabolites, or lipids. And we term these analyses proteomics, metabolomics, or lipidomics. And you can just see from these photos through the years the things that we've worked on, anywhere from hardware development to improve the sensitivity of mass spectrometry measurements, or liquid chromatography system development to improve throughput and efficiencies of separations. Software, you can see some of these graphics here, as well as actual instrument development, new instrument development. And the cartoon that you see here of an eye mobility spectrometry instrument was commercialized by Agilent a number of years ago, and is now sold on the market as the 6560 IMS QTOF system. And then the latest technology that has been developed in the lab are the structures for lossless eye manipulations, and that is now going through the courting phase of commercialization with the relevant companies there. And you can, of course, see the instruments here that just gives a little snippet of the capacity that we have here. And it's the capacity to perform large-scale studies is actually one of the greatest strengths of the lab, both through the Integrative Omics group and our partnership with the EMSL as a user facility, and having upwards of 40 instruments and collecting data annually for around 50,000 samples, and those numbers just continue to increase. So I don't mean to insult anyone's intelligence, but maybe not everyone has heard of the omics or is familiar with the different flavors of the omics, but I always like to include this slide as a little bit of background as well. It just gives you a pictorial view of the omics spectrum, all the way from genomics, which is the first omics, arguably, all the way through proteomics, and then the different flavors of small molecule analyses. These are either metabolomics or lipidomics or glycomics. If you roll all of these together, including anthropogenic molecules, then we can call this exposomics. But for the purposes of what we were trying to accomplish with the UDN Metabolomics Core, we were employing metabolomics and lipidomics, and sometimes you would get data from these other molecules along with the targeted focus on endogenous molecules. But we were implementing these in untargeted paradigms, meaning we weren't trying to perform absolutely quantitative analyses. We were trying to be as comprehensive as possible to measure as many molecules as possible, but that might then provide some insight on the diseases that these individuals were suffering from. And then with a typical omics experiment, you either take a single data set or a omics data set integrated with other omics data sets, and you rely on some pretty sophisticated informatics and bioinformatics tools and methods to then develop a model that might explain the phenotype of interest that's being observed. So this is the paradigm that we're implementing here with the UDN Metabolomics Core. And our goals are shown here. I'm not going to read them. The important parts are listed in bold text here. And we never quite got around to this third goal of performing structural characterization of novel molecules. We had more or less enough work with our time in the UDN to focus on this first one, the second one, and then the third one, essentially providing guidance on what metabolomics could offer to a particular case, generating the data, and then doing our best to interpret the findings in the context of other data that was available for the same patient, such as gene mutations. Our workflow looked like this. This is actually a figure from our proposal that we originally submitted to the NIH in our application to be the Metabolomics Core for the UDN. And this didn't really change through the course of the program, and we included this figure in the UDN manual of operations, and I think it's still there. But essentially, a case comes into the UDN, we discuss it among the larger UDN team, which, as Dave pointed out, was pretty big, 18 institutes, a lot of investigators. But when we go to these meetings with the UDN team, they're all very collaborative, they're all very interesting discussions, and you leave them usually wishing you had more funding and more resources to try and contribute to a solution to the diseases that are being presented. But once we decided collectively with the UDN as a consortium that metabolomics was warranted, then we would ask the referring physicians if there was an existing hypothesis or not. If not, then we performed what we considered a full metabolomics workup, which included metabolomics and lipidomics analyses. If there was a hypothesis, which usually very rarely there was, just due to the nature of the disorders being studied, then we would take a targeted approach, and we would collect the data, and maybe that data would lead to a new hypothesis. Very often not. And then we would communicate that data back to the UDN referring laboratory and work together to see if there was anything that we could come up with that might point to a reason for that person's disease. So I mentioned just briefly, our approach was to measure metabolites, which we refer to as metabolomics analyses, or we would measure lipids, which we refer to as lipidomics analyses. And these aren't, when we perform lipidomics, for example, we're not performing a blood lipid panel. We're looking at the very detailed molecular composition of the lipidome, all the phosphatidylcholine, all the phosphate ethanolamines, so on and so forth. to try and understand or get as much granularity in the molecular profiles as possible. So one of our biggest challenges with the UDN was sample power. So you've got individuals being referred to the consortium that for all intents and purposes are the only ones that are presenting with a particular suite of symptoms. And so we had an analytical challenge here. How can we derive meaningful information from the analysis of a sample from someone who's enrolled in the program when we've got essentially a single data point? And so this is maybe a little bit less of an issue for target analyses when you're actually generating quantitative information, and you can refer that quantitative information back to your favorite resource of normal values. But when you're performing semi-quantitative analyses and you're looking at just fold changes with p-values associated with them, it becomes difficult to trust anything that is determined to be statistically significant if you've got data essentially from just a single individual. And so what our approach was to try and establish a reference database of normal values. And part of the challenge that we had with that approach was knowing how many normal individuals or healthy individuals did we need to study to populate this reference library. And so our approach to determine that was to perform a power analysis on historical metabolomics and lipidomics data generated using the exact same analytical pipelines that we would use to study individuals enrolled in UDN, and using some pretty typical assumptions of the data from mass spectrometry based omics studies. And so in performing that type of analysis, and one key point here to point out is we were assuming that we might need as few as 10 to upwards of 200 to comprise the reference group. And we're also assuming that we would have as few as 1, or maybe wouldn't say as lucky as 10, but as many as 10 for the disease groups. So we're assuming an unbalanced study design in that analysis. And so through that process, we found that we would need 80 to 120 individuals that were healthy or otherwise not known to have metabolic disease in order to detect a twofold change in over 90% of the molecules that we could measure with the following metrics here. And what we were able to achieve in that sense is listed here. We were able to obtain plasma, urine, and CSF samples from the numbers of individuals shown here. And I put these asterisks here because it's difficult to find an appropriate set of samples. We did a lot of emailing to people in our network to ask if they had access to people that fit our criteria. And fortunately, a lot of these people that we contacted said yes. And fortunately, again, they were very willing to share the samples. And so I just thank those individuals down here, some of whom are at OHSU, for contributing these samples. And some additional criteria here for these samples were that we're trying to match the typical demographics of a person enrolled in the UDN. That's more or less 50-50 distribution in between males and females. It's a majority of individuals under the age of 18, and it's a majority Caucasian. And the real challenge was to get samples from individuals under 18 who were disease-free, because usually you aren't studying or you're not taking clinical samples from children unless there's a presumption or a suspicion that that they're suffering from something, and then they've gone to a metabolic clinic, for example, to have samples taken and analyses performed. So for the samples from children that we were able to obtain, we can't guarantee that they don't have some type of disease, but we're pretty sure that they were not suffering from a metabolic disease that would then influence the metabolomics and lipidomics data. So what we had in mind of how this background data set could work looks like this, which is a paper from Sarah Elsey's group at Baylor, in which they used untargeted metabolomics that was generated in a collaboration with Metabolon to study individuals with known metabolic disease. And so this would be things like fetal ketonuria and so forth, the very well-known inborn errors of metabolism. And you can see that with a normal background of data from some number of individuals, the normal values usually fall within plus or minus 2 or plus or minus 2.5 z-scores. And then with the metabolic disorders where you have accumulation or depletion of certain molecules, these really stand out next to that reference population. And so this was what we had in mind in performing our analyses. And I should say that with our effort to establish a background population, we had this challenge of an N of 1. The challenge that we encountered through the course of this program also wasn't just due to an N of 1 in terms of the number of individuals that were available with presenting with a certain disorder. It was the fact that they, very similar to the UDP, they would go into the UDN clinical sites for a 1-week workup, and they would have one sample drawn, one sample of blood, one sample of urine, one sample of cerebrospinal fluid if their symptoms warranted drawing CSF. And that was the only sample we ever got. And so we would have liked to have had more than one sample, repeat samples from the same individual, on and off medications, on and off special diets, and so forth. But we weren't able to get that all the time. And Dave will show a slide a little bit later that really shows the potential power in obtaining a repeat samples from the same individual. So what we would do then was to, again, establish the reference data set, analyze the patient data, normalize the patient data against the reference data set, generate the z-score plots, perform traditional statistics, calculating fold change in p-values, and then communicate that information back to the referring UDN clinical center. So this is a figure of a representative result from a particular patient, and I like to present this example in all of my talks about metabolomics because I feel it really highlights some of the technical challenges that are just still faced using the technology today. So this is just a portion of the polar metabolomics data that we were able to obtain for this individual. This patients are usually referred to as probans in the vernacular used in UDN. And this list goes on, down below what can be displayed here, but I just like to show this portion here. And so through the course of analyzing the data and plotting it against the background that we generated, our background data is shown here in gray, and you can see it's very similar to what was shown in that paper from Sarah Elsey, and that normal values are around 2 to minus 2 z-score. But we found a very interesting metabolite that was 4-fold, 4 z-score increased in the patient, the patient's mother, and the patient's father. And we thought we were really onto something with this particular molecule. And then we scroll over here to the left, and we realize that it's unidentified metabolite 67. So it's something that's not matching the reference library that we have for annotating metabolomics data. So it's very exciting yet very, very frustrating. And we don't know if this molecule is this high in these 3 individuals because it's determined genetically, or if they all had the same breakfast, or if they got new carpet in their house and the carpet is off-gassing and they're accumulating something due to that new carpet. And so very frequently we have to sort of forget these types of findings and just move on and try and interpret the data that we actually can annotate. But I'll point out here that with metabolomics, typically less than 10% of what you detect in the instrument can actually be assigned a chemical structure, and that's just where we are right now at the state of the art. And the reason for that is because of how the analytical paradigm works. We being the metabolomics community, purchase authentic reference compounds from companies, We get these in pure form, we run them through the instrument, we collect the experimental information, we put it in a database that becomes our reference database. Then we take a complex sample like blood plasma, we run it through the same instrument, we collect the same data, and then we look for where the data matches up, and where the data matches up, we assume we've made a confident identification. The problem with this paradigm is that less than 1% of what we know or suspect is chemical space, you can actually get in pure form. So there's a real limitation right now in the field of metabolomics with this paradigm. And I just put in a plug here for our new capability that we're developing at P&L that we call standards-free metabolomics. And this is being accomplished under an NIH funded core, as well as some internal efforts. And we're trying to change the paradigm where we replace this top half of analyzing compounds in pure form, and we rely instead on advanced computational methods, either machine learning or quantum chemistry, to predict how these molecules are going to behave in the instrument. And then we can populate really comprehensive reference libraries based on these predictions. And so theoretically, we only need to know the two-dimensional or three-dimensional chemical structure of the molecule in order to generate the reference information. And so at this point, we'll get to the lessons learned portion of the talk, and I'll turn it back over to Dave.
You're muted, Dave.
That might be a good thing. Yes, here we go. Okay, we're back. Okay. So lessons learned. Well, we learned a lot. I especially learned a lot as a clinical metabolic physician going out to PNNL and getting the shock and awe tour of all the instrumentation out there. It was kind of like learning mass spectrometry through a fire hose. But there was also some more practical things that we did learn, and I think are important lessons from the standpoint of not just the utility of metabolomics in reference to a diagnostic tool for rare and undiagnosed disease, but I think are also lessons that are important for people who are considering using metabolomics as a research tool, and in that it's very important that you have an appropriate design if you want to get anything meaningful out of this. And there's a lot of things to control that aren't necessarily intuitive, and that is also another reason why it's great to have this collaboration with PNNL, because those folks, Tom and his colleagues, and are experts in the field with this, and so it helps a lot in this whole process. But anyway, jumping right ahead here. So first off, metabolomics. There are big differences between using it as a research tool versus a diagnostic tool. If you're doing metabolomics as a research method, it's pretty straightforward. You've got two or more groups of subjects, or maybe you've got people who are fasted and not fasted, or you've got a mouse and then a knockout mouse. But you have groups that you're comparing. And so first off, you can have a larger sample size instead of one individual, and thus you get obviously a lot better statistical power. And then you can also control for things, or at least it minimizes the impact of things like the diet or how long they're fasting. And the other thing that's key is that you don't really need a normal, because you're not taking a one individual and saying, well, what's different about this person? We use metabolomic type things all the time in the clinical evaluation of patients with inherited metabolic diseases. But as Tom showed from that one figure from the folks down at Baylor, we're looking for things in which these metabolites, the abnormalities are statistically extreme outliers, Z-scores of 8, 10, 20. So it's easy to find those things. But when you're looking at things that aren't so extreme, it's much more difficult. And again, if you have a good sample design, you really don't need to know normal, because you just need to know what's the difference between your groups. Yeah, contrast that with the use as a diagnostic tool. You have one patient and maybe one sample, as Tom mentioned, and then this idea that you need a normal because you have to, or at least a reference group because you have to make a comparison. And so approaches that we used and things that are important to consider in improving the utility include analyzing samples from multiple family members, both affected and unaffected. That gets you back in the idea of having two groups if you've got affected and unaffected relatives, and then multiple samples from the patient as well. And then the other key is getting good information on that particular patient. Are they having symptoms now or are they not? You know, there are episodic conditions where you have abnormal metabolites when they're symptomatic, but not when they're asymptomatic. And again, diet, medication, supplements. So it's really key to have all that information. And we did incorporate the collection of this in the sample requisitions. But you know, a lot of these patients are on a lot of things in terms of supplements and funky diets. So it becomes very difficult. The next figure is just an illustration of what you see and the power of having multiple samples. This was from an individual and their mother. So the patient labeled P here and the patient's mother, M. These are, what this is showing is a heat map. Anything in red has a z-score that's positive, so that's an increase relative to the reference group. And anything in blue, there is decrease relative to the reference group. And what this data that is shown here is from 294 individuals. And each one is represented by a vertical column. And then within that column, each red dot or little square is a different lipid. So this is a lipidomic profile. I can't recall whether this was a positive or negative lipids, but it's not important for this purpose. But what you can see is that there's a lot of variation across the entire group here. But in particular for this patient, again, we had multiple samples, which was very helpful. And I think it's really clear right here in this cluster of samples from the patient and the patient's mother that you can see that there's this group of lipids that were consistently decreased in this particular patient and increased as well. Thus the red here for the increases and the decreases. Some of these were also found in the patient's mom. And you can see at multiple time points that these things persisted. So here's in 2016, here's later that year, and then in 2017. So there's a lot of benefit to getting multiple samples. But again, that's really difficult in the context of a routine diagnostic workup. So there's other things that I think are important that are really going to be necessary before metabolomics is what I would refer to as something ready for prime time medicine. And that is improving what is normal. Because again, a twofold elevation, you know, when we think about z-scores, 2 to 3-fold elevation or a z-score of 2 or 2.5, statistically, it maybe is not all that exciting. But on the other hand, what if you may have situations where you have a handful of metabolites that are elevated and then a corresponding group that are decreased. So the idea of getting into things such as patterns, ratios, and correlations among these hundreds of metabolites, as opposed to just utilizing z-scores. And that sort of thing, of course, requires better bioinformatic methods. So instead of just looking at a heat map like we were just looking at, using better bioinformatics to really try to identify what is a normal profile and what's an abnormal profile, and then ultimately, hopefully incorporating additional information such as phenotypic data or even diet and microbiome information into that to help interpret with all this data. I think the other thing that could be very helpful would be the utilization of tissues, either like biopsy samples or cultured cells, because then you can control the environment, the physiologic state based on the culture conditions. And again, you can do provocative things. You could grow them in the absence of glucose or do whatever. And then animal models, because again, you can control the environment for animal models, and then you have cultured cells, you can control or compare those to cells cultured from animal models. And all of those things would be very helpful in terms of a diagnostic thing, but it should be pretty obvious that this is not something that's going to be used just in your average clinic. I mean, this is really kind of a research undertaking, and that was the role of the UDN. Now, one of the big goals of the UDN was, in fact, to try to figure out how to roll this out to more common usage, but that's a big challenge. So just to kind of summarize, well, I would say that we all agreed it wasn't really quite ready, metabolomics, that is, as a diagnostic tool. On the other hand, if you get patients with identified conditions, even in this context of like a single patient or only a few, it may be actually quite helpful for understanding pathophysiology of these conditions. But there's also the other issue, and Tom kind of pointed that out in showing his slide where he showed that the patient and both parents were outliers for a particular unidentified metabolite. Is how do you know when an abnormality is really a diagnostic finding of some significance versus an incidental finding? And I just throw up a couple examples of metabolic diseases, which are really just biochemical abnormalities that don't have any associated clinical symptoms. And yet when you run things like organic acids or acylcarnitine profiles, you'll see all these abnormalities that look like it could be a disease. So, and that's the most important summary and conclusion, is that working with PNL was a lot of fun. I'd highly recommend it. And again, I think one of the great things about it was, as I mentioned, I'm a clinical metabolic disease specialist, primarily, and there are no clinical people at PNNL, or at least I didn't work with any. And so it's a great collaboration because you have this incredible strength out there, and yet they have all these tools that are really begging for clinical applications. So it was a lot of fun, and I think it was a great time. And then just some acknowledgements. These are big teams. So of course we have Tom, who's not even listed here, and Bobby, Joe, and Katrina, and Jennifer were the primary folks at PNNL who worked with us, but it was a large group of others. And then myself, and as I mentioned, Melissa Hendel, and Matt Brush, and JP Gordine also. And here's one of our meetings. There's Jennifer and Katrina and JP and Melissa here. And then this is the whole, we always met, or most of the time we met in this hotel in downtown DC, and here's a big picture of the whole group. So at that point I will stop, and we'll open things up for any questions that folks might have.
Thank you both. That was excellent.
Yeah, that was really interesting. We've got a few questions in the chat and also some comments. The first question early on was from Dan Marks, and he wants to know if the network also looks for intoxicants, environmental contaminants, and poisons.
There's no specific toxicology screening, if you will, but some of the folks at Harvard developed a very detailed environmental, call it an exposure profile that was used not consistently in all patients, but in some patients to try to collect information about potential exposures. Because I think, if I can assume, Dan, what you're thinking about is the idea that, a disease may be rare because it's, someone happens to be exposed to something, or when we see 3 people in a family, that have something similar. But there was not a toxicologic screen, if you will, incorporated into the routine analysis of the patients. No.
Thanks, Dave. Can I be so bold as to ask a follow-up question?
Oh, I suppose.
You'll tolerate it. Thank you so much. You know, the other question I have is the issue of disease-modifying genes. So sometimes we find a family who has, you know, 2 children with extremely variant phenotypes for the same mutation. And I'm curious if there is anyone in your consortium who's interested in the concept of disease-modifying genes, or, you know, why is it that the phenotype can be so completely different with the same primary mutation in a known disease-causing pathway?
Well, pardon my blunt answer. I'm sure there's a lot of people interested in that. But what I would say is that, again, there was nothing that was directly coming out of what was going on in the UDN to try to do that. Again, it's a little bit like the problem of metabolomics, right? You know, if you have 10 patients with something, you might identify a pattern. If you have 2 siblings, you know, you can do their whole genomes and you can figure out what they do and don't share. But then going from that and figuring out, well, which is the modifier gene and not. So, I mean, I think it's a very good question. And at one level, there was sort of a look at that in terms of in patients where no obvious diagnosis or etiology was found through, say, sequencing techniques, the idea that there could be epigenetic phenomena. Now, this wouldn't be modifier genes per se, but it would be what you might think of as a non-inherited genetic abnormality for some reason or the other. So either through RNA-seq or methylation analysis, things of that nature, to try to tease things out. But yeah, I mean, I think that the other, though, is really, again, something that requires, at least at current time, you know, a number of patients, so you can do that sort of mapping within pedigrees and between families.
So Sarah Goslin has her hand up, and I think she and Ruth have similar questions, and it's related to this whole topic. So do you want to go ahead and...
So I have a different question than Ruth that I was going to ask. I have a question primarily for Tom. So I don't know if you want Ruth to go first, and then I can answer
you
it.
can go ahead, and then Ruth can go next.
Okay. So Daniel, I have a lot to say about your question as someone that works with a known gene mutation with gajillion modifiers and relatively few patients. But that's a different thing. I have a question mainly for Tom and sort of on the research aspects of the metabolomics research, because I found it really interesting how, you know, kind of looking for known metabolites that distinguish between disease phenotypes and healthy phenotypes, you get, you know, undiagnosed, unknown metabolite 67 or whatever the exciting thing was. But you kind of touched on this machine learning aspect where you're trying to learn patterns in metabolites from an unsupervised standpoint. I was wondering if you've been able to do any sort of feature reduction so that you're able to identify not necessarily a known metabolite, but a known feature or subset of those metabolites in disease patients versus not, or if you're looking into that.
Yeah, so the figure that I showed, that machine learning, was not in the context of pattern recognition, but it was in the context of taking a chemical structure and predicting, say, its retention time in the liquid chromatograph, or how that chemical dissociates inside the mass spectrometer. But to the point of your question, yes, we have been using machine learning, and I don't do it myself. I work with Bobby Jo Webb Robertson, who's on the call.
Okay.
And she's applied it in a lot of our studies, primarily in the context of type 1 diabetes.
Okay.
Where there's genetic information, there's metabolite information, there's protein information, there are diet surveys, and you know, you get this very nice descriptive disparate panel of markers. So it's very useful in distinguishing, say, someone who's genetically predisposed and probably on their way to developing autoimmunity all the way to the point of type 1 diabetes. I'd say the disadvantage to those approaches is that the panel that you get often isn't related in terms of a molecular pathway or a signaling pathway. And so then you got to start digging in and really trying to suss out why those, why this metabolite is a part of the panel, why this gene is a part of the panel, and so forth. So, depends on what you need, for your study. Do you need a descriptive panel of markers, or, do you need a pathway level and a mechanistic understanding? Two different types of analyses.
I guess I feel like if you can understand the features that are distinguishing, again, sort of the computational features. So like it could be, drug activity or something, or sort of whatever chemistry things that you were saying. Not a chemistry person, but you can sort of look for patterns in that and see and then sort of dive in and see what they have in common. But it sounds really cool, and obviously, like I do work with Bobby Jo a little bit, so I can ask her more about type 1 diabetes on another call. Thanks.
I highly recommend Bobby Jo.
Good, I'm glad.
Ruth, I think the floor is yours now if you have a question.
Hi. My question was in regards to people who have a known variant in a known gene. And I feel like there's several like rare and extremely rare diseases like this where they've known for a long time what the variant was or variants, and they know the gene, but they really don't know anything more. And I always feel like these patients are in some ways at like a huge disadvantage because people are like, you already know the gene, you already know the variant, and it probably does this. And then some people don't look into it, whereas if you're truly undiagnosed, people have all this interest in like diagnosing it. So I'm curious if your network considers these people, and I feel like it would be such a cool resource for these people because they have so much data accumulated.
Well, I mean, I guess there's sort of two ways that there has been demonstrated, to try to look at these. I mean, one is the animal model core. And so particularly with novel genes or genes of uncertain function that are found by genetic means to be associated with symptoms, developing a fly model or a fish model, et cetera, has been used, because there you can try to tease out things in terms of mechanisms, pathophysiology. The other, as I mentioned, in terms of the metabolomics, I think that's another area where there could be a lot of utility, and that is not trying to identify necessarily what the gene is or what's going, but what's unique about that particular patient. Perhaps even bringing in the proteomics as well, because you're trying to understand, well, yeah, they have this gene and there's this mutation and there's something different, but it's not intuitive. So trying to understand what's different about this person versus their sibling who doesn't have the mutation or something. And then I think the omic things, such as proteomics and metabolomics, or potentially transcriptomics as well, getting at the idea that you maybe have some expression differences as well that are underlying these phenotypes. But it is not something that was a big component of the UDN. That was, the UDN was looking for that gene and that variant. But in terms of understanding pathophysiology, other than potentially making an animal model to try to tease these things out, that's kind of where things went. But you make a good point that, great, now I have a name for my unknown disease because I got a gene, but that's it, right?
I think the problem in like autoimmunity and autoinflammation is that it really takes that environmental trigger. And so you breed a mouse, which we did, and then they have no disease, and then you're like, now what? So yeah, you kind of have to use the patients in that way. So
yeah, and I think it really takes the people who have really deep expertise in that particular area. So such as your area, the autoimmune and the inflammatory kinds of things, to address that, because it really becomes a research question.
So I think we probably need to wrap up here. I just do want to point out that Ann Lewis has put a comment in the chat that there is a macaque resource at the Oregon National Primate Research Center that's a genotype and phenotype library. They've got a data bank and tissue from a lot of these animals, which could potentially be another resource on the one hand to even check some of those animals to see if they have mutations that show up or that are some of these things associated with diseases, but it's not clear how the phenotype expresses itself. And they've got comprehensive sampling of all kinds of tissue. So I just wanted to make sure that that point got communicated. And we're well after 4 o'clock, so I want to thank the speakers for such an interesting talk and all of you who came and engaged with questions and comments. And we will see you again in a few months, where we're going to hear from another collaboration between OHSU and PNNL. So thank you all.
Thanks for having us.
Thanks.
Thank you.
Thank you.
Bye.
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 Bye, everyone.